What is the difference between genetic engineering and selective breeding? Have you ever wondered how we can create new plant varieties or animal breeds? The answer lies in two fascinating techniques: genetic engineering and selective breeding. Let's break down what sets them apart. Selective breeding, also known as artificial selection, is a method that humans have used for thousands of years. This process involves choosing parents with specific traits and breeding them to produce offspring with those same traits. For example, farmers have used selective breeding to create different types of corn or dog breeds. This method relies on the natural genetic variations that already exist within a species. However, it can be a slow process, as it depends on the natural reproduction cycles and the traits available in the gene pool. On the other hand, genetic engineering is a more modern approach that allows for precise changes to an organism's DNA. In a laboratory setting, scientists can directly alter the genetic material of an organism. This can involve inserting genes from completely different species, such as bacteria or even viruses, into the organism's genome. Because of this direct manipulation, genetic engineering can produce results much faster than selective breeding. It is not limited by the existing genetic variations within a species, allowing for more innovative traits to be introduced. Now, let's look at some key differences. First, genetic engineering is faster and more precise. It allows scientists to introduce specific genes from any species, while selective breeding is slower and relies on traits already present in the species. Next, the methodologies differ. Selective breeding uses natural breeding processes, while genetic engineering involves direct manipulation of DNA in a lab. Both techniques find applications in agriculture. However, genetic engineering is often associated with genetically modified organisms, which have sparked debates about safety and environmental impact. In the realm of emerging computing technologies, particularly biointegrated computing, understanding these techniques is essential. Genetic engineering can be likened to programming in computing. Just as software developers write code to achieve desired outcomes, genetic engineers introduce specific genes to create organisms with particular traits. This analogy highlights the potential for biological systems to be engineered with the same precision as software. In summary, while both genetic engineering and selective breeding aim to modify organisms, they do so through very different methods. Selective breeding relies on natural variation and is a slower process, whereas genetic engineering allows for rapid and precise changes at the genetic level. Understanding these differences is key to appreciating how we can harness these techniques in agriculture and beyond.